All right, good afternoon, everyone. It's my pleasure to introduce the speaker for today, Alán Aspuru-Guzik from Harvard, and his talk is going to be entitled Learning from Nature, Bacterial Light Harvesting Antennas, Perspectives for New Organic Materials for Solar Cells. I thought I'd give you a little brief bio about Alán. He's currently an associate professor of chemistry and chemical biology at Harvard. He started there at 2006. He got his undergrad in chemistry from the National Autonomous University of Mexico in 1999, and then he went to Berkeley for grad and postdoc, and he got his PhD in physical chemistry under Professor William A. Lester, and was a postdoc at Martin Head Gordon's group at Berkeley from 2005 to 2006. His research is at the interface of quantum information and chemistry. In particular, he's interested in the use of quantum computers and dedicated quantum simulators for chemical systems. He's also studied the role of quantum coherence in excitonic energy transfer in photosynthetic complexes, which he may touch on today. He's developed methodologies for studying the spectroscopy of molecules in nanoscale environments, and he and his group recently developed the density functional theory for open quantum systems. He leads the Clean Energy Project, a distributed computing effort for screening renewable energy materials, and I think he's definitely going to be talking about that today. And some of his awards, just quickly. In 2009, he received the DARPA Young Faculty Award, the Camille Henry Dreyfus Teacher Scholar Award, and the Sloan Research Fellowship. In 2010, he received the Everett Mendelsohn Graduate Mentoring Award and received the HP Outstanding Junior Faculty Award by the Computers and Chemistry Division of the American Chemical Society. In the same year, he was selected as a top innovator under 35 by MIT Technology Review magazine. So with that, let's introduce Alán.
Oh, thank you, Mark. It is very, very nice to be here. Thank you, Mark, for the invitation. Can you guys listen to me in the back? Yes? Well, Mark already told you what I work on in my group. I just wanted to briefly show you the areas and how they are connected, especially because today there was a Nobel Prize associated a little bit with this area, of the idea of how to use quantum devices, how to control quantum devices to actually simulate systems. And in my group, we're very interested in the prospects of actually using quantum simulators or quantum computers to actually simulate molecular systems. As Mark told you already, we have had an effort on thinking about how to think of the electronic structure of molecules in dissipative environments, say a molecule next to a plasmonic nanostructure, or molecules, say, in these two settings, which are what I'll be telling you today about. I'll be telling you first, in the first part of the talk, my work on this area that actually Mark coined Excitonics. So I'll tell you what Excitonics is and how does it fit in the context of photosynthesis. And then I will tell you some work in my lab on organic photovoltaic materials. And at the very end of the talk, I'll try to connect them with recent work of a group from Japan. So that's more or less the direction, and gives you an idea of what my group does. We are theoretical chemists. We don't do any experiments yet, but hopefully we don't do experiments. We'll be very bad at it. So let me just begin about the idea of excitonics. This is in the context of a center at MIT, Harvard, and Brookhaven that actually my group partakes, and is one of my most exciting projects. The way to, I think, describe excitonics is actually thinking about two of its neighboring fields, the field of photonics and the field of electronics. And if you actually think of these two fields, and you want to think about how to move particles or quasi-particles from one place to another, actually they are quite advanced. For example, in photonics, if you want to, for example, move a photon from place A to B, you can actually use an optical fiber or a waveguide, or say a photonic crystal, and you have very good control of how to move a photon from point A to point B. Similarly, in the case of electronics, as you know, we can actually pretty much control electrons and holes and move them anywhere we want, because we actually have Coulomb forces to play with, and we can actually create very nice transistors, right? But excitonics lies here in the middle. An exciton, as I'll show you in the next couple slides, it's a bound electron-hole pair in the context that we're talking about in these soft materials. It's a bound electron-hole pair that only leaves very little and actually is charged as neutral. So therefore, it's very hard to move it around from one place to another. And so that is the goal of our Center for Excitonics, to think about how to move around excitons, how to manipulate them, how to understand them. And what I think is quite interesting also is that nature actually has mastered excitonics, and in the case of photosynthesis, has done great things with excitons, because that's the way, say, plants and bacteria harvest sunlight, right? So today I'll tell you a little bit about our work in that area of how photosynthetic light harvesting organisms work. And later I will tell you about how to think about artificial devices that actually also do light harvesting. So let's just begin with green-sulfur bacteria. So let's just describe this organism, Chlorobaculum tepidum, green-sulfur bacteria. It lives in many places, hot environments, for example, Yellowstone, places like that. It also lives in the bottom of the ocean, even 1 kilometer down. And when it eats around those environments, it actually receives 10 to the minus 4 watts per square meter. Okay? So that is a solar irradiance that translates actually to 300 photons over the entire cross-section of these bacteria per second. And these photons are actually also low-quality photons. All the good photons were already harvested by the bacteria and algae that are above in the ocean. It actually is receiving near-infrared photons. That leads to a very, very slow duplication. Perhaps once in the time I've been at Harvard, this guy has duplicated once, okay? So 3 to 26 years, okay? One duplication at this irradiation. And how does nature cope with it? Well, in this particular case, the reason this organism is quite interesting to us is because what nature decided to do is to actually compensate for this very low intensity of light by using an extremely large photosynthetic antenna complex that uses up to 250,000 bacteriochlorophylls per a few reaction centers. And you can actually see these huge bags of full of pretty much a liquid crystal of bacteriochlorophyll molecules near the photosynthetic membrane. So this is the structure of this organism. So why don't we try to compare it, for example, with a saguaro, which is, I come from Mexico, so I like to show you this Mexican plant here, that actually receives 10 million times more irradiance, and therefore does not necessarily have an antenna that is as large as this 250,000 bacteriochlorophyll antenna. It suffices to have about 200 chlorophyll molecules per reaction center. And probably it has evolved to do something more close to photoprotection rather than this guy that probably doesn't need sophisticated photoprotection mechanisms when it receives so little photons. Okay? So it's more interested in actually harvesting, presumably. So for anybody interested in the biology of this organism, here is basically the different components of this very early photosynthetic organism. It only has one reaction center. It uses menaquinone as its electron transfer or electron carrier. And we'll be not focusing over the very, very slow process of converting this harvested light into actually biomass. But we'll be discussing actually this intriguing antenna complex, base plate, and FMO quantum wires that we're showing you that harvest the sunlight from these 250,000 bacteriochlorophyls all the way down to the reaction center with about 98% efficiency or so. So that's also amazing because this particular light harvesting part of photosynthesis is actually very efficient. Overall, photosynthesis is not necessarily the best idea to actually, for example, get energy out of, say, corn, for example, for doing ethanol. That is probably less than 1% efficient, solar to biomass. Okay? But what we're talking here is just basically the idea of light harvesting, which one in turn can use, for example, for catalysis, or can use it for organic solar cells or for any other application. So technologically, it's very interesting to understand, to me at least, how does this organism decide to be so efficient harvesting light at so low conditions. You can even think about it as a photodetector if you want. Okay. So let me show you a video that one of my undergraduates made as part of her undergraduate thesis project that does not contain any simulations. It's more like a public video that shows you how this photosynthetic organism actually works. Okay. So this is not the oil spill, but it's actually we're coming down the bottom of the ocean. This is her artistic representation of photons. Okay. And in this particular case, she chose to go only 100 meters, but again, you can go up to kilometers even. And here are these green objects that represent green-sulfur bacteria. And the photon is going to hit these bags full of chlorophylls. They are full of these concentric rolls, of which I'm going to only show you one of them. Okay? This is the actual atomistic structure, and this is her artist's representation of this excitation moving around this photosynthetic role, going through this Fehn-Matthews-Olson complex in a second towards the reaction center. So here's our dramatic turn, entering the Fehn-Matthews-Olson complex, and then the reaction center. Okay? So I'm going to summarize to you a little bit of a technical result that actually was very important for the field, an idea of how the energy transfer happens in these systems. Okay? Actually, one of the creators of this is here, at least, Masoud Mohseni. Actually, I see him sitting there. He's now at MIT. And this is the idea of environment-assisted quantum transport, which is how, based on ultra-fast experiments I'm not telling you today about, we now think about energy transfer in this complex. So this Fehn-Matthews-Olson complex is some sort of excitonic wire. Now I'm showing you was one of the monomers of a trimer that connect this antenna complex to the reaction center. Okay? And the efficiency of this FMO complex, again, it's above 98%, actually comes because these electronic excitations actually do not transport like this, just hopping between one side or the other, but more like this, so to speak, like a quantum wire, so to speak, delocalizing and interfering between different chlorophylls. Okay? So this idea of environment-assisted transport that we posed is the idea that the high efficiency of the system is actually somewhere in between the quantum and the classical regime, where if you're very, very classical, the efficiency is very low, and if you're very, very quantum, the efficiency is also only about 80%. Or at the natural noise levels of the bacteria, we'll discuss later how the noise of the bacteria actually allows the exciton to transport. Actually, you get the best possible transport. So this is kind of like our tutorial picture of it. You get, if you had no molecular vibrations, your excitations will be delocalized over different chlorophylls. But the molecular vibrations at room temperature actually fight against that and localize the wave function. And it is the interplay between the two. What I'm showing you here is actually the population on each one of these chlorophylls. It is the interplay between these two that actually allows to have a high efficient energy transfer. Okay? So this is as technical as probably as I will get in this talk, but I wanted to show you this video, just to give you an idea of some of the research that we do here. But today, what we'll be discussing is, okay, given the fact that we have noise and given the fact that we have delocalization, how do they play in this antenna complex, which is the new results that are coming out of my group this summer? How does the antenna work? And they are kind of preliminary. Some of them are published, some of them are not. Okay, so I'll be showing you them in a little way. But let's come back to this exciton. We talked about excitonics. What is an exciton? Well, an exciton is a molecular excited state that can be delocalized over one or more molecules. In this particular case, I'll be talking mostly about chromophores such as chlorophyll or porphyrin and things like that. It is exciting for us when they delocalize over one or more, so let's just talk about the delocalization mechanism. If you have a second bacterial chlorophyll close to it, they can interact electronically and generate one of these splittings due to this coupling between these two different excited states. These are the ground and these are the excited states of these molecules. That the coupling is about the order of room temperature and therefore generates a splitting about of the order of room temperature. And we can think of these delocalized states on this particular dimer that I'm showing you here as an example as a symmetric state and an antisymmetric state that will be two excitons that delocalize over these two chlorophylls. In photosynthesis, usually you have delocalization over, say, a few, in the FMO complex 3 or 4, and in this antenna complex I'll be telling you about perhaps a few hundred. So that's more or less where we are going. So that's what is an exciton. How does an exciton move? Now let's localize an exciton in a particular bacterial chlorophyll, and let's first form it by actually interacting with sunlight and exciting the molecule, say, from its highest occupied molecular orbital to its lowest unoccupied molecular orbital, as a cartoon of the usual more complicated many-body excitations here, and then find an acceptor somewhere. Okay? And this exciton, therefore, will be transferred from this donor to this acceptor by a resonance, by an exchange of a virtual photon. When the energy levels of these two are brought to the same level due to the fluctuations of the environment, then we get an exchange. This molecule will be de-excited, or this group of molecules will be de-excited. And this group of acceptors, or this single acceptor that I'm showing you here, will be excited. And that is what we like to call resonance energy transfer. And there are two regimes usually for energy transfer. The one that people have been mostly focusing on until recently is this idea that oscillatory effects, for example, due to quantum coherence, decay extremely fast, And therefore, you can think about it really as a kinetic process that goes from the donor to the acceptor. Here we have the populations of these two chlorophylls that at a very large time are equal between the two. But at the very beginning, you start localizing one and slowly transfer to the other. But if the actual oscillations if the actual decoherence rate or the loss of coherence in the system is actually comparable to the energy transfer rate, you will actually see these little bits here. Okay, you will see some sort of coherence that actually might change the transport mechanism. And this idea of incoherent versus coherent has been a huge discussion in the literature, which I'm really not touching on today, only to show you that coherence, many recent experiments have shown that coherence might play a role in this energy transfer. Okay? So what today I want to concentrate on is how does this antenna work? And if we can learn something from that antenna for, say, thinking about photosynthesis, artificial devices. Okay? So let's just first look at the structure of this antenna complex. There was about a 15-year debate of how this antenna complex looked like, because it's actually a liquid crystal of bacterial chlorophylls that self-assembles inside of this bag, so it's kind of quite disordered. So they had to generate a triple mutant structure of this bacteria that actually created this andouille sausage of chlorophylls here. Okay, I'm just maximizing this little roll here. And you can actually see that in this particular structure of the triple mutant, they were able to use cryo-electron microscopy and nuclear magnetic resonance to actually obtain this atomistic structure. Okay, so this is the atomistic structure that was invented by the group of Hoptegrud. Just came out in 2009, and we set out to actually think about, well, if this is the antenna complex of the organism that harvests sunlight most efficiently that we know of, why don't we try to understand how does it work? Okay, so that's where we are. So this is basically a cartoon of our of all the pieces of our atomistic model, the atomistic model that we have. Okay, so let me just take you through it. I told you quickly that there were actually three components. I'll be highlighting them here. The actual chlorosome, which is this roll of bacterial chlorophylls. The bag might contain many of them sometimes, grabbed concentrically. Okay, so here I'm just showing you three of them. Then in the wall of this liposome bag, there's actually a set of proteins that actually also contain bacteria that form this two-dimensional sheet that is called the base plate. Okay, an energy transfer goes from the chlorosome to this base plate towards the reaction center, the FMO reaction center. So it's a very, very spatial energetic funnel. Okay, excitations here are actually higher energy than the FMO and the reaction center. And somehow you need to have a near unity efficiency of energy transfer from here to there. This is a moment that I like to invoke my analogy of a stadium. Okay, so compare this room, which perhaps has a few, maybe 100 people, or even less than that. And that's more or less the number of bacterial chlorophyll molecules that you need to actually generate an excitation on, and transfer the excitation, say, all the way down to Mark over here in the center. Okay, so if you even think about it semi-classically as a hopping process, it's not so hard to think about say that lady with the laptop in the back harvesting sunlight and bringing back the sunlight all the way down to Mark. But if you have 250,000 chlorophylls and you have to concentrate it all the way down here in the reaction center, right? You need to think about a stadium, say the Olympic Stadium in London, and any person anywhere, right, harvesting sunlight and bringing it down to the center with 100% efficiency. So that's what I want just to think about. So many bacterial chlorophylls here all the way down to the reaction center. So why do we think that this chlorosome has this shape? Okay, so why, as a chemist or so on, I might be able to speculate, why does this chlorosome have this beautiful shape? Okay, so there's a couple reasons. The chlorophylls in this system are actually, and I don't have the slides, I forgot to add a couple of them, but they are aligned in what is called a syn anti-aggregate. The transition dipoles are actually organized in a cylindrical fashion with very, very strongly coupled chlorophylls that actually allow for this guy to talk to light as a collective. Okay, so it's like a big collective dipole that talks to light, so to speak. And if you want to think about a dipole that talks to light, why would it be cylindrical? Well, because then no matter what orientation does the light come from, it will be a good antenna. Think of the car antenna of your car. Okay. So we actually have done some electromagnetic simulations with collaborators at Northeastern University that show that this seems to be the case. Okay? So you have this radial orientation of transition dipoles that we think is important. Second, as I will show you from some of our simulations, excitons delocalize really fast, and also from experiments. These excitons delocalize really fast over pretty much the entire roll. Okay? So therefore, you get coherent transfer from between them and amongst them. Okay? Internally and also between them towards the base plate. Okay? And then, because the exciton is delocalized over the roll, you get an enhancement of the energy transfer to the base plate. Bob Silby here from MIT, and later Seth Lloyd and Masoud Mohseni have worked on this idea of super transport. Okay? The idea that if you have an excitation that is delocalized over more than one donor, and you have an acceptor that actually has more than one chromophore as an acceptor, the rate is enhanced as the number of donors times the number of acceptors. Okay? As I will show you the results from our simulations, huge delocalization here, in turn, deals to huge enhancements of energy transfer due to the delocalization of the exciton. After the exciton is transferred to this base plate, then there's two-dimensional diffusion of the excitation that needs to find this lonely FMO reaction center, and in turn transfer the energy to it. Okay? So this is how we think this organism is actually working. Okay? And our goal is to actually have a systems-level study of all the components and see what we can learn from it. So in this recent paper, Takatoshi Fujita from my group, Jenny Brooks, and Semyon carried out a simulation on small models, say of the order of hundreds of thousands. In this particular case, I'm showing you 600 bacteriochlorophyll molecules of this roll to try to understand what is the diffusion constant, say, for example, of excitations and the energy transfer mechanism in this system. Okay? When light hits this roll, you don't start with an excitation localized in a single site. It delocalizes over a particular region of the roll, and then it transfers from there downwards. Okay? But to actually think about the exciton diffusion, it's actually good to actually start with a population in one particular site and say calculate the diffusion constant. Okay? So this is a result from a single trajectory. Okay? You see here the populations between the different sites of this roll oscillate back and forth. Probably about 200 femtoseconds you actually went around it. Okay? And there you see it. Okay? So this is the results of a calculation whose all of its parameters came from quantum chemistry or atomistic simulations. Okay? And then that fits a quantum master equation for those of you that want to know how we did this. Okay? And in this particular case, this ensemble simulation, okay, is just an average of many of these trajectories. Okay? So, sorry, the ensemble will be the quantum master equation. This will be stochastic type Schrodinger type equations. Okay? So we also do master equation simulations on this as well. I'll show you one of them later. So this is the result of a stochastic simulation. This is the average over the ensemble of a few hundred or thousand trajectories. I think this is 1,000. And here is again the delocalization. So we posted our paper on this archive ePrint server, and then about a week later, this paper appeared in the Journal of Chemical Society, an ultra-fast spectroscopy paper by Donatas Sigmantas that actually shows, by means of this two-dimensional spectrum that the details I'm sparing you of, their observation, they associated with a delocalization over 100 femtoseconds over the roll. Okay? So the comparison of theory to experiment is not so bad. We're not simulating this signal, but we're actually having these direct simulation results. Nevertheless, the timescales agree. So the atomistic model seems to have some validation from experiment. And if that is the case, then this fast delocalization over the roll is actually meaning something in comparison to this experiment. So we're very happy about that particular agreement. So now, this is actually, so I do have the structure. This is actually a picture of the structure of the roll, a little bit in more detail. You have these bacteriochlorophyll molecules stacked in these cylindrical rolls. You get very strong coupling along this direction, and a strong but not a strong coupling in this transverse direction. Okay? So you might think that transferring one of those directions might be more useful. In this particular case, it would be the direction that takes you towards the base plate. Okay? And it turns out that the base plate, from all the biological information we have, seems to be stacked here in this particular axis of the cylinder. Okay? And because of this strong coupling here, depicted in blue, compared to this weaker coupling in the z direction, actually excitons diffuse way faster in the direction that seems to be matter to nature. So we also like a lot this result. There is an artificial saturation here. This probably is more like so, because we really simulated a cigarette butt of a chlorosome. Okay? We don't have too many bacterial chlorophylls in this direction. Therefore, our simulation actually saturates. But this gives you an idea, again, of the fact that you get faster exciton transport in the direction that seems to matter for this organism. Okay? So now let's look at some of our simulations in different components of the system. So these are the timescales, very preliminary. Okay? They might be changing in the next few months. Of the different aspects, right? So I told you the one that we published already is the delocalization along this cylinder. Okay? That's about 200 femtoseconds. So you might ask yourself, what is the time scale of going from the interior cylinders to the exterior cylinders? Our preliminary number is about 400 femtoseconds. Okay, so this is a result of that simulation. This is now a quantum master equation simulation using the same parameters. And you see here the little concentric rolls light up when you start an exciton in the center of this concentric set of chlorophylls here. Okay, we get energy transfer from here to the base plate, from the rolls to the base plate for about 1 picosecond, and then again about 1 picosecond towards the reaction center. These are very, very fast timescales because the exciton lives, up to nanoseconds, about 1,000 times more than that. And seems to be the case that at least in our preliminary simulations, it takes only about a couple picoseconds or so to start, say, in a place in a roll, and just, boom, shoot all the way down to the reaction center. Okay? So this is the first, so to speak, systems level study simulation of this system. Of course, we want to have a longer base plate, concentric rolls, larger calculations, and so on. And this is a calculation that needs to be in a supercomputer, and we are actually running it as we speak. These are very recent results. Okay? But this is more or less what we have right now as our first baby simulation of the entire light harvesting of the system. Okay? So I just wanted to start the talk with this, just to show you an idea of how this bacteria actually is doing its business, okay? And then discuss, well, what were the things that we learned, okay? So we think that the large-scale and efficient exciton transfer in the system comes from the strongly coupled chlorophyll molecules that make a well-defined molecular aggregate, right? So if you want to harvest sunlight, perhaps the self-assembly of these chlorophylls is crucial to be able to actually induce this exciton delocalization that in turn induces fast energy transfer. Okay? And now the questions that we can think about it is, can we learn from this anything to try to design a better organic solar cell, which is what I want to discuss later, in the next part of the talk. And also, can we even think about it in principles like this for organic electronics? And these are questions. I'll tell you some of the directions in my group towards answering some of these questions. So now let me just tell you about, switch gears a little bit, and tell you about organic photovoltaics, how we screen materials for it, and then I will tie things back together at the end. Okay? So this is kind of like the first world picture of organic photovoltaics. You see this lady that needs to charge her iPhone. Like me, my iPhone is always running out of battery with all these apps. Okay? And therefore, she has this beautiful bag that actually charges the iPhone, right? And this is this defunct company, Konarka, that actually produced these organic solar cells roll-to-roll here in Massachusetts. There's about order of 10 other companies that are still in business trying to do the same thing. This is a view that actually I wanted to show because it actually comes here from MIT as well. This is my colleague Sheila Kennedy here in actually architecture. And she's working with these companies to think about other uses for the 2 billion people that actually have no electricity in the world. So those were solar cells in a rooftop. This is a Huichol Indian in Mexico using organic LEDs for actually organic lighting devices to actually read at night, apparently, right? And actually, instead of having that high-tech bag, she's actually working with the Huicholes to actually make solar ponchos, okay? So this is pretty cool, right? This is kind of like a good use of organic electronics, I think, because it doesn't matter, or photovoltaics, it doesn't matter how much you spend at the very beginning, right? The first few watts are very important. So here you have this guy actually charging his phone, right? So this is about a $20 device as far as I understand, okay? So this is actually, I think, a cool use of organic photovoltaics, probably the first place where probably they will impact the world, I think, Okay? So with that motivation, I'll show you the typical plot that everybody that works in solar shows you. And the reason I show it to you is because the people in organics like to show it to you, because it's basically the progress in different solar technologies. I was born in 1976, so you can see that silicon solar cells were quite crappy back then. Okay? But now, they are saturated about these efficiencies. And we know they are very cheap, for example, because of China's subsidies and so on. Right? So they're very, very good. But if I was an assistant professor in chemistry, I would not bet my career in thinking about improving these solar cells because they are actually quite saturated. Whereas the world of organic electronics, when I started at Harvard, was in 2006 or so, was with solar cells that were as crappy as when I was born. Okay? But now they're about 10% efficient. And one of these 10% efficient solar cells or so comes from Mitsubishi, and I will be talking to you about it at the end of this talk. Okay? But you can actually see all these different solar companies, including the defunct Konarka, coming up with different high-efficient solar cells in a very little amount of time. Okay? So my group, when we started in 2006, started thinking, what can a computer do to actually help us find new materials to get to these 10%, which we already got with a very few materials. Okay? So for that, I'm going to show you a diagram that actually Roel Sanchez, which is here in the audience, actually, he's now at Bosch, made for one of our grants. I still show it because it's very clear. This shows you how these organic solar cells work. We're going to have a donor material. In this particular case, we picked one that is not a very good donor, but nevertheless, it has a lot of interest recently again. So this is material called pentacene, but any organic polymer, for example, or chlorophyll molecules will do this. You can generate these excitons, these electron hole pairs that diffuse to this interface with an electron acceptor, which is usually a Fullerene derivative. It doesn't have to be. And the energy levels in this particular case are aligned in such a way that you can get enough free energy to dissociate this electron that falls down to the cathode, and the hole floats to the anode. And there's many, many issues in these organic photovoltaic materials that people tackle. Where I was just going to tell you about the idea of trying to find materials for them. And at the beginning of this talk, I told you about one issue that we've been thinking about. How do excitons diffuse? Seems perhaps that nature teaches us something about it. But to make a good solar cell, you actually need to think about how they are formed, how they diffuse, this very important problem of the interface, how the holes and electrons move around, and the interface with an electrode. Okay? So it's a very complicated scientific and technological challenge. So let's look at another cartoon video that my student Lauren made of how this works. Okay? So we are entering now this blend. Usually these things are a blend, say, for example, of this polymer and this Fullerene. And this is Lauren's artistic representation of an exciton, electron hole pair, diffusing in the polymer, reaching the interface with the electron acceptor. So here we have the dramatic rotation around the Fullerene, showing now the dissociation, right, of the electron going into the Fullerene and the hole diffusing back into the polymer. Okay? So this is kind of like a artist representation. Actually, it's funny that the actual atomistic configuration comes from a simulation from the Jenny Nelson group in the UK. So actually, the actual atomistic placement of these Fullerenes and these polymers is actually, so to speak, realistic. And here you see the challenge of this field, right? There is this blend that comes through between these aluminum and indium tin oxide electrodes, and a lot of the properties come from this idea of the microstructure, right? How can you actually have a good blend of the donor and the acceptor in such a way that excitons reach the interface fast, dissociate, and jet transport to the electrodes? Okay? So the assembly is a big aspect of it. But another aspect is materials, right? Ultimately, the materials will give you what you want, which are the parameters for this famous solar cell characteristic curve. This is the voltage at zero current and the current at zero voltage that define the limits of your solar cell. The performance of the solar cell is this curve, that the more square is better. That means the experimentalist made a better solar cell, has less leakage and less problems. Okay? So it's called the field factor. And ultimately, as theoreticians, it's extremely challenging because what we want to do is actually get this short circuit current and open circuit voltage from, for example, theory, and actually try to find what is the best material to do that. And luckily, if you're interested, for example, just on the voltage, right? You can do that very easily because you know the energy level of the acceptor material. In this particular case, we're going to use this Fullerene as an example. And you have oxygen as another limiting parameter, right? You don't want your molecule to be oxidized. And therefore, one can show, Marcus Sharber showed in 2006, that this is more or less where you want to be in terms of the energy levels of your donor molecule. So the question that we are trying to answer is, what is the actual donor molecule that actually will be good for this? Okay, donor molecule or polymer. So this is the gap, the difference between the HOMO and LUMO levels. And I'm going to plot it against the LUMO in the next plot and show you this model by Marcus that actually shows where would you like to be 10% efficient solar cell. It happens to be here. Okay, so you want a band gap of about, here, about 1.6 or so, and a LUMO level around -4 and so on. And the question is, what material does that? Okay, and then the answer, can be difficult, right? Because if I ask all of you, my organic colleagues, what is the best molecule here, for example, for an organic electronics device, it will be very hard to actually find out. For example, what is the best Hall conductor here? Does anybody give me any suggestions? No? Which is very hard, right? Turns out to be 7, right? I know that, but it's very hard to know beforehand, right? So that's one option. The other option is, of course, picking one of them and telling our collaborators to synthesize it. And I work very closely with a group of Shenam Bao, I'll show you later, on trying to screen these materials. And it takes about 1 human year to synthesize and characterize this. And finally, the other way we can do, another possibility is theory, but even theory, right? Theory is not as great if you do it the way we do theory right now, which is do one molecule at a time. So Schule, Johannes, which is actually present here, or Roberto, if you gave a molecule to each one of them, maybe they can do a molecule a day if they are very hardworking and do drink a lot of coffee. But it will take a lot of time for them to actually screen a reasonable number of materials if they do it by hand. Okay. So we try to get another strategy, which is learn from the people that search for extraterrestrial intelligence. Okay? And when I was an undergraduate in Mexico, I was a geek, and I actually downloaded this thing called SETI at home, which is a beautiful project that actually convinces people to Fourier transform the sky by giving them a nice screensaver. Okay? So these people are actually downloading the screensaver and helping the SETI project find aliens. Okay? I quickly switched to actually folding proteins, okay, because I thought that was more efficient than searching for extraterrestrials. But I really like this concept. And between then and now, nobody was doing quantum chemistry in a distributed fashion until I became assistant professor. We're talking about 7 years. So I decided to actually start one to actually find organic materials. Here are some of the people in the team at Harvard. I see some of them here. Roel, who is now at Bosch, is here. He started it. Johannes is here as well, and others perhaps from my group. This is the Stanford team, the QChem team, and IBM. So let's try to see if we can figure out who is who. This is a quantum chemistry company, and this is, as you know, a big computer corporation. So what team is this one? You can probably imagine by seeing Johannes over there. This is the Harvard team. Okay, diverse and excited. Okay, this is the Stanford team. These are organic chemists, as you can see from here. The QChem team are our people that develop the software package. They really look like programmers, you can tell. Okay? And they are awesome, by the way. And finally, IBM corporate people. Okay? So all of us team together, right? And why do we team together? Because we're actually doing something like this. Another video by Lauren. Okay? Excuse me for showing so many of them. I love them. Right? So the idea here is actually searching organic molecules with volunteer donors of computer time around the world. Okay? So you, me, anybody, when your computer is actually watching the Daily Show or something, right? You can actually use it to actually, when it's actually doing almost anything, use the idle computer time to actually screen for materials. Okay? So roughly each one of these computers every 12 hours will be screening one of these materials, okay, to find out new solar cells. Okay? And just today we probably have gotten 35,000 or so more molecules in our database. You know. So maybe the lucky person will get a T-shirt from us or a coffee cup if they find the right solar cell. And the reason IBM is involved, of course, because we have to have a website. We have to make sure there's no viruses on our software. We have to modify QChem and so on. So it's a huge enterprise. There's about 500,000 people that donate computer time to this project, out of which, of course, way less number is active. And we have to compete with other projects that IBM hosts as well. Okay? So that's the reason why there's so many people. So this is a project that actually uses this screensaver. Okay, this is how it looks. Ours doesn't look as cool as the SETI at home, but I think it looks pretty cool with our leaves here. And it's going to give you an idea of the scale of this project. It is equivalent to having our own supercomputer. Okay, we have computed the same amount of time since the dinosaurs started sorry, dinosaurs, excuse me, I'm an idiot. I'm sounding like a creationist. Since the mammoths were around, okay? Since the mammoths were around, okay? I'm checking my time right now, okay? Yeah, so since the mammoths were around until now, okay? So there were still mammoths when we started computing. If you had one computer and the computer was running from then to now, okay? So this is actually another way of thinking about theoretical chemistry. You have to actually run a calculation every 2 seconds, okay? So just think about the issue that, of course, as good as Johannes is, he cannot prepare an input file every 2 seconds without the help of a computer, right? So that's why we need a lot of framework to actually do this. And we collaborate actually under the umbrella of the Stanford Global Climate Energy Project, which seems to be like an equivalent project to the MIT EI, in the sense that it's actually a project to actually invest in energy in the university. We get a subcontract to think about with Shannon and Michael about this project. This is our cover on the project. You can read this paper to learn more about it. But the idea, of course, is that we get a lot of ideas from our synthetic colleagues and we send them back materials. Okay? So really, we want to do that. So this is the slide of Johannes that I will go quickly telling you how the project more or less works. Okay? We started with 26 fragments that we combinatorially make about a library of 10 million molecules. Roberto did that. Then we screen it with quantum chemistry, but also with chemoinformatics. And when we use chemoinformatics and quantum chemistry, we benefit from modern tools, for example, machine learning, which are, for example, commonly used, say, in pharma, to try to find out what is the best material. From that, we use the cyber infrastructure of the Clean Energy Project. Put stuff in a database. I'll tell you about the database in a few slides. Now this is where we're putting a lot of effort, as you can see from the faces here, in data mining and calibrating this stuff to be able to actually release our top molecules. And then I want to just skip our complicated diagram, okay? Just to show you that we come back to the experiment again and give prototypes to the bio group. And we already have done that once. And we sent them recently a new list of molecules that is synthesized and it helps us do our search. All of this is automated using the wonderful Python programming language. Okay? What is in yellow there is what we're actually working on. And what is in, what is working already, but we're still improving. And what is in yellow here is what we, are moving on to next. It gives you an idea of the scale of this project. That's why there were so many phases involved. Okay? So here, for those of you that care about organic electronics, these are the 26 building blocks that we fuse and link to actually do this. Okay? Here they are. Okay? And after we do those combinatorial stuff, I'm skipping a lot of stuff in the interest of time, we do machine learning. And just to remind you what machine learning is, let's just think about the face of this ugly guy here. Okay? So let's imagine I want to predict his face. And of course, machine vision is a very important project, right? I want to have my iPhone recognize faces, for example, right? So the way it's done is, for example, think about features in this face. For example, you can see that there are 2 eyes, right? And that, for example, that between them there is a nose, right? So you have something called a weak classifier. For example, 2 circular things next to each other. That would be like a weak classifier. If you see 2 circles, perhaps that's an indication of eyes. But he could be a pirate or have an eyes clothes, right? So that classifier might not be enough for an eye, so you might want to look for a nose in between or big ears after it and so on. So in this particular slide, the classifiers are slightly different. Basically, how similar is this guy to, for example, these pictures here, right? And perhaps you will imagine that you will weight these 2 pictures more than these 3 pictures over here, right? And it is this concoction of combinations of weak classifiers by combining them just by weights that give you these strong classifiers that will be able to tell you if say, this phase is, recognize this phase. And it's very similar to an organic material, right? I want to find out if there are descriptors on it. For example, it's homo-lumo levels. I just showed you one descriptor that is pretty good. But there might be other more subtle descriptors that will tell you this material is actually a good or bad for solar cells, right? And that's more or less the kinds of things that my group is doing right now. Of course, not with these phases, but with materials and with descriptors from pharma. And what we're building a lot is this type of idea of classifiers to find out the materials. So that's the idea. So now I'm going to show you some of the results, right? These are the HOMO, highest occupied molecular energy levels, of different levels of theory that we used in this screensaver, okay? So for those of you chemists, you might know what this means. If you don't, what is interesting is that the main criticism of our project is that how can you do absolute prediction on this? Because all the levels are, all the levels are very different, right? And what I want to tell you is that when you calibrate with experiment, all these, as I'll show you in a second, all these islands actually collapse into the same data. Okay? So you need to calibrate with experiment. And I'm showing you now calibrated data between of the HOMO and LUMO energy levels of these molecules. Okay? Of course, this is ugly, so I'm going to show it to you in a more nicer plot. But we still have an issue here, right? I need to actually go to probably the first time I've seen a plot with a logarithmic number of molecules. So we have about tens of thousands of molecules here, showing you the dynamic range between the HOMO and LUMO of these 4.5 million molecules that I'm showing you here. Gives you an idea of the dynamic range of these molecules that we have searched with the screensaver. And now I'm going to show you calibration. These are the different methods without calibration to experiment all over the place. As you compare them to this calibrated data, that of course these islands are slightly different depending on the level of theory, but they more or less lie on top of each other and gives you an idea of the confidence that we have on an electronic structure. Okay, so there's still a lot of work to be done, but at least when you calibrate, all these islands go to the same area. And what I'm showing you here is the same gap versus LUMO plot I was showing you before. So let's go back to that gap versus LUMO plot. Let's try to see what lies here in the 10% solar cell region. It's here, okay? And you see that we have a few thousand molecules, about 15 to 25,000 molecules in this region of 10%, okay? For all the levels of theory, they more or less look the same. That's what I'm trying to show you here. And the point is that it's actually a very, very small percentage, about 0.3% of all the molecules we explored. And what we really like is that most of the molecules we predict actually lie over here, okay? Where most of the organic molecules that people have synthesized in the world lie, about a 5% organic solar cell against PCBM. So even if you have a lot of organic chemistry intuition, you probably are picking at random from this set of molecules and roughly landing in this area. So computers might be able to help us the same way computers have helped the pharma to guide synthesis, to tell us new ideas about the fragments that are in these molecules. Not necessarily give us the molecule, but give us guiding principles to be able to synthesize more molecules here in the 10%. Okay, so this is our database. We promised, because of the Materials Genome Initiative, that we're going to launch it in 2012. So we're working very hard to launch it, hopefully before the year ends. Or, if we break our promise, early next year. But here it is. You can search by data fields, et cetera, et cetera. Here is our beta version. And I'm just going to show you really quickly a video of the kinds of things you will be able to do when this data is out there. Okay? You will be able to, for example, search the database, okay, using these tools from pharma. This is a particular tool called Vortex. And actually click, for example, here, HOMO versus open circuit voltage. You cannot read what I'm telling you what it is. And shows you the relationship between HOMO and open circuit voltage over these 100,000 molecules or so. And then what Leon was doing in this video is actually showing you he can actually plot this Sharper plot on this small sample of our database in real time to you there. And he can actually go ahead and fish molecules. So it is this leveraging from the tools from pharmaceutical industry to organic materials that we're trying to strive towards. And this year we promised also we'll release our, or early next year hopefully, if not too late, our top 25,000 molecules. And every year they will be very different because we learn better how to do machine learning. So we're learning how to do learning. And we're also screening more materials, right? So every year we'll try to give this experimentally some guidance of what are the patterns that are more common on the molecules that are more efficient in this search, okay? But this is more or less who we are in this project. So I wanted to end now, in the last 5 minutes of this talk, connecting the two subjects because actually Mark Baldock here actually raised my attention to this paper, which is a paper that he told me seems to be the basis of this Mitsubishi solar cell, right? As far as I know. Right? It's one possibility of what is the Mitsubishi solar cell. Okay, so he sent me that paper about a year ago or half a year ago, and I'm very interested about this paper because that paper actually talks about stacked porphyrins, and we talked about the chlorosome before, right? The chlorosome is a bunch of stacked porphyrins, and turns out that the trick that is done in this paper is actually have a variant of this or these benzene porphyrin molecules that is soluble. And that upon heating leaves, basically by a retro Diels-Alder reaction, loses these organic ethylene groups here and becomes insoluble and crystallizes. And they are able to manipulate the crystallization process in such a way that they can make actually beautiful pillars of self-assembled bacterial chlorophyll molecules. And in this paper, it's about a 5% organic solar cell. Okay? So, and this is actually the acceptor, which is also, they claim, important to have these silicon-containing groups here to actually make it self-assemble better. That's what they claim. So the question is, well, you have these self-assembled chlorophyll molecules, and then these fullerenes. That's the picture that they show in their paper. This is the self-assembly of these chlorophylls in this pattern that actually resembles the pattern that I showed you before in the chlorosome. Okay, so that's the connection I want to make. So perhaps this solar cell is working well, and we want to study that because of its possible connection to what nature did. So it will be kind of cool if this is true. Okay, I'm just telling you my thinking as of the day. And this is their picture of a self-assembly of the columns of acceptor that they are able to make. Here is actually pictures of the device. Okay, by microscopy you can actually see here. This is the one that has you can actually see here the columns of these chlorophylls filled in between by this fullerene. Okay, these are different views of the device. You can see here the different layers. Okay, and again, this is a respectable solar cell for the organic community. Presumably improved by in the same way for 10%. It might not even be the Mitsubishi solar cell, but what matters is that this seems to be a very good direction that we want to think about, and that perhaps connects the two fields. Because so far my group has been working on the photosynthetic side, thinking of exciton transport, then thinking about the materials, but it's not so clear how to, for example, screen, for example, what would be the what I'm thinking. So what I mean is, what would be the next direction would be, for example, to screen for the best porphyrin variant, and then try to have it self-assembled in a similar way as, say, the chlorosome, right? So I just wanted to share with you more or less what we're thinking, okay? And really, the last slide of this talk is just telling you that I told you that we closed the loop with Shannon one time. We didn't really close it in the organic photovoltaics domain. We closed it earlier. And actually, Roel here was a very important part of that team. We closed it earlier thinking about a simpler problem, which is charge transport in organic materials. We screened variants of the thiophenated organic molecules, okay, to actually come up with this molecule that to date is the second best whole mobility material in organic electronics, that according to Samsung, they came to tell us that. And the story then is, can we use, basically the reason I want to end with this is, can we use this idea of, for example, coherent delocalization, which is our current explanation of this very high whole mobility on this very, very rigid organic crystal, to not only optimize all the components that I told you before. We want to optimize the material, which implicitly contains the energy levels, for example, the HOMO and LUMO levels, which is what we've been working in our clean energy project. Optimize the transport, as we've been thinking about in photosynthesis and these organic solar cells, of excitons in the case of photosynthesis, and on holes in terms of these organic electronics. But if you wanted to really make an organic solar cell working, you want to have all those components working together in the same device. Okay? And that is still challenging, and that's why we only have very few cases of getting about 10% organic solar cells. So I hope this talk gave you some perspective of the type of questions that we are trying to address in my group, and more or less our state of the art. Some of the things I showed you are preliminary results. Some of them at the very end are even speculations. But I just wanted to share with you how we are today, what we're thinking today in my lab. And with that, I want to thank the people in my group. Here are the current graduate students and postdocs, as well as undergraduate students. These are the undergraduate students who have worked in my group all over the year, and former group members, collaborators, and funding sources. And with that, I want to open for questions in the remaining time I have. Thank you. Any question?
Yeah, on the chlorosome structure simulations, which are very impressive, it wasn't clear to me to what extent those structures were based on experimental structures of the chlorosome from electron microscopy, crystallography, so on and so forth. Could you comment on that?
Yes. Sorry, I tried to give a general audience talk, so I didn't talk about that in detail. So the story is like this. We take the structure that was published in that paper of 2009, which is obtained from cryo-electron microscopy and NMR. Okay? And then we make a subset of it, and we do molecular mechanics calculations on it. We jiggle it in the computer, okay? And we get those, and we do then on top of that quantum chemistry calculations that tell us an idea of how the electronic energy levels are fluctuating on these bacterial chlorophyll molecules. These are the parameters then that actually feed our stochastic simulations. From that, we get a autocorrelation function of this gap that in turn informs our quantum models of transfer. And we have validated that in the Fena-Matthews-Olsen complex, which is the smaller component, getting pretty much agreement with experiment. Here, it's hard to actually really compare directly to experiment, other than this time scale, right? So we are trusting our method as much as we can, but it was validated on the Fena-Matthews-Olsen complex. So that's more or less how we do the simulation. I don't know if that answers the question.
Basically, so what's the resolution of the experimental structure before you do the jiggling to relax it and so on?
What is the resolution in the cryo-electron microscopy?
Correct.
I don't know what is the resolution of the combined NMR and cryo-EM out of the top of my head. But you can read the paper, but I started from the protein data bank structure my colleague sent me. I can tell you the resolution might not be as good as you think, because this comes from a very noisy system, right? It's a self-assembled, roll. And because the tails of the chlorophylls that they gave me are actually too much on top of each other, to the point that we are working very hard on relaxing this structure. So it means that, they place the chlorophylls, but the tails are not necessarily well placed. So yes, it's not a perfect structure, but we're working a lot in relaxing it. So that's more or less what it is. The resolution, I don't know if it's as close to that of a protein or something, but we can read that in the paper. Luis?
Are you planning to take into account aggregates or clusters of that sensitive molecules you are finding in the big screening?
So you're saying combining? Yes. So he's asking me if I can combine and study clusters or aggregates of the molecules.
Like the fed absorption.
Yeah, of the molecules.
Will occur from absorption, and the mobility of excitons will occur in aggregates.
I will be very interested. Yeah, of course. We screened first we screened first the small oligomers in the clean energy project, right? And then we calibrate with experiment, which is another leap of faith. Once we have those, right, we want to bring them back to this level of theory that we did for the chlorosome. Okay? That's why I wanted to show you this, perhaps disconnect the two first parts of the talk, right, and try to connect them at the end to tell you where we're going, to put them together and really create like stacks of these. For example, in the case of porphyrins, try to find what's the best porphyrin, right, and then stack them together and see if we can simulate the exciton transfer in them or the absorption. As you say correctly, aggregates absorb light differently than single molecules. I hope that's where your question is leading to. So, there's another question back there? It was the same question? Good. You guys are in synchrony, right? You're coherent with each other. By the way, there's also Masoud Mohseni here. I mentioned you already, right? I'm going to make sure all the players that did this work are here. Is there any other people with another question or concern?
I have a quick question before
yes.
we finish up. There's going to be refreshments up there.
That's great.
For everyone. Did you make a solar cell out of did Genome make a solar cell out of the Da2T?
Oh, we discussed that. Yeah. So Mark about a year ago told me, did Shannon make a solar cell out of the Da2T? It seems to be good because the band gap is probably okay and so on. I think the issue is that they made the crystal, they measured the crystal for the organic electronics, and they moved on. So, and I think, yes, perhaps we should come back to it and maybe do a solar cell of it. Who knows how good it is, and but that was in the plans, and then it just was dropped by our experimental colleagues. But yeah, you could in principle make an organic. Maybe I should not call her. She calls me with many demands for new calculations, so I should demand the solar cell, right? As a joke, right? We're good collaborators, so it's like, hey, Shannon, have you made the solar cell? Have you made the solar cell? Maybe she makes it. Yeah, so yeah, we haven't made a solar cell out of it. Just the organic electronic device, and I don't even know if we have more material. Yeah, or they have more material. Yeah.
All right. Well, with that, any further questions? You can confront Alán in person at the refreshments. Thanks.
Okay. Thank you.